Welcome to Scigest, your fortnightly serving of digestible science from plant and food research.
Kia ora koutou katoa. Welcome to Scigest. I'm your host, Hilary Ireland, and today we have another episode in the Science Life for Me series. Today we have the pleasure of speaking with Dr. Julie Bloomaert, scientist in the seafood production group. Kia ora, Julie. Welcome to the podcast.
Hi, nice to be here.
Before we learn about your science journey, can you tell us about your role at Plant and Food Research and a bit about your current research?
Well, I've been at Plant and Food a little bit over 1.5 years now as a Post-doctoral Scientist, and my work is related to a data science fund from MB, looking to use data science to improve aquaculture outcomes. So I focus a lot on the genomics, so sequencing DNA of snapper, and sometimes things like their microbiome and things like that, to see how we can help them grow better and faster. And that's also working together with other groups that try and apply things like machine learning to the genomics data.
Great, that sounds really exciting. Yeah. So do you have a strong data science background?
I actually started in the lab with more of a molecular biology and I guess pure genetics background. But I guess these days everything is kind of heading more and more towards being computer-based, and that's sort of how I ended up where I'm at.
Yeah, I couldn't agree with you more. Everything does tend to head towards a lot of data analysis and data science.
Yeah.
So where did your interest in science begin?
Basically, growing up, I lived sort of in the countryside and surrounded by nature, so I think I was always quite captivated by that. And also things like looking up at the stars at nighttime. But I also grew up in my parents' bakery, and my dad always made a point of explaining to us sort of every step of the process of whatever he was doing. And I think that's not something that everyone sort of thinks of as a science, but it definitely is.
Absolutely. You can tell a good cookie from a bad. There's definitely an art and a science to good baking.
For sure.
Great. Well, with so many varied interests in science then, so how did you ultimately choose what to study at university?
I guess like a lot of young kids interested in biology and in nature, I initially wanted to be a vet, but just kind of a few things happened in life that ended up putting me off of that. And then I didn't really know what I wanted to do, and I considered being a pilot for a while, and I think I still want to learn to fly one day. Mm-hmm. But in the end, I sort of decided that I was quite interested in genetics, and I had the opportunity to shadow a genetics researcher for a day in high school, and that sort of set me down that path, and went to uni for genetics at Otago, and then went overseas for a little bit for PhDs and postdocs, and yeah.
So was it a high school subject where you first came across genetics, or was it looking at variation in nature? How did you discover genetics?
I've never thought about that, actually. I think it was a high school class, yeah. Mm-hmm. Yeah.
And how did you get to shadow a genetics scientist?
It was just something that I expressed interest in, and one of my teachers organized it for me. I can't recall if I specifically asked for it or not, but yeah, it was a really cool opportunity.
Excellent. Sounds like a great teacher.
Yeah, definitely. Yeah, I still email her sometimes. She did at one point ask via my sister when I was going to finish my PhD, which was a little bit of a low blow at that point.
Oh, really? Okay.
Because I really wanted to finish it. It was getting to be time, and you know when everyone's asking you when you're going to finish and things like that, and then your sister writes you, "Oh, our high school science teacher asked when you're going to finish your PhD," and I was like,
I don't need that right now, thanks. Worst timing. Yeah. Right, well, so you did a PhD then. Can you tell us about your experience of your doctoral studies?
Yeah, I actually sort of was told through my bachelor's and honours studies that if I wanted to come back to New Zealand, that the best plan would be to go overseas at some point. So I figured why not do that during a PhD? And I actually headed over to Europe to start a PhD, and it didn't really go very well with my supervisors. I think we just didn't really click at that point. And that was about a year in that I made the call to leave that PhD. I did try and find other supervisors at the same university, but it didn't really work out that way because there were just a lot of practical concerns like access to the organisms that I was working on and things like that. So it would have been too awkward and weird to try and work that out. So then I knew that I wanted to still do a PhD, and I stuck around for a bit and visited some other labs. but I couldn't find funding immediately. So I came home, and it was around Christmas as well. So it's just kind of like dark European winter time, and you don't have a job. And it's like, yeah, I may as well come home. And then I ended up working in a cheese factory for a while in Nelson while I was still looking for other PhD opportunities. And then that took me to Austria, where I ended up doing a PhD looking at genome size change in rotifers, so little zooplankton that coincidentally we now feed to our fish. So kind of, yeah, little links in the research, even unintentionally.
With your first PhD, would you mind sharing some lessons learned from that experience?
Yeah, I think it's sort of important to understand yourself and what you need, I guess, and to be able to advocate for that. And I think that's probably not something that you learn easily. Mm-hmm. You kind of either have good mentorship and you learn that way, or you kind of just are thrown in the deep end and have to figure it out for yourself.
Do you think you took those lessons forward when you were looking for your second PhD?
Yeah, I think so. And I was really open about it as well when I was applying for another PhD. And that led to some, really good discussions about, I guess, needs and expectations around mentorship and work and communication as a PhD student as well. And I think that's probably a really important conversation to have with your potential PhD supervisors anyway. But yeah, I guess sometimes if all sides are a bit inexperienced, like if you're someone's first student or second student or something, it might not be so apparent to the supervisor that that's a discussion they need to have early on.
Absolutely.
Yeah.
That's some great wisdom right there. So how long did you work in the cheese factory?
About 6 months, so actually not very long. But yeah, it was really fun, and it kind of felt like science in buckets, and it was delicious at the end, so no complaints really.
So you still feel you had a hand in science when you were working there?
Yeah, definitely. And I'm still, I think, also really interested in things like fermentation, even though I don't use it in my work. I've sort of had a go at, I guess, like everyone else through the pandemic, sourdough and home brewing and
Yes.
things like that. So it's definitely been a nice little interest on the side. Yeah.
Nice. And so your second PhD, that all worked out nicely and you finished that up in 3-4 years?
Yeah, it was 4 years, so technically not to plan because everyone plans for 3 years, but I guess we all kind of know that doesn't happen anyway. I spent about a year, I think, in the lab trying to get a karyotyping method to work, so to be able to count chromosomes in these rotifers, but sometimes I think that's more of an art than a science, like getting the really fine details of that kind of protocol sorted. So in the end, we had to flag it, and I guess that was another lesson to learn when to give up on something.
To give up, yes.
Yeah.
Was it tricky working with the zooplankton?
Yeah, it was actually, because especially for karyotyping, you need cells that are dividing so that the chromosomes are condensed. And that's usually like in developmental stages, and in these zooplankton that happens in a really tough shell. So you have to be able to crack that but not damage the cells, and that was one of the sort of main sticking points. But even after that, it was really difficult, and it turns out the cells were really fragile, so they would tend to just break up, and then you have chromosomes floating everywhere instead of something in a group that you can count. So yeah.
So with that hurdle of the karyotyping, you still achieved what you set out to achieve with your PhD?
Yeah, I think we had planned a little bit more experimental work and lab work for me, but in the end, with the genomics, we just had so much data that I ended up with enough work from that for my PhD. Sure. So.
Great. The stage after completion of one's PhD can sometimes be a very uncertain and challenging time. How was this time for you?
I'd say it definitely was very uncertain, because I finished my PhD at the end of 2019, and we all know what happened at the start of 2020.
Yes.
So I was lucky enough to have a postdoc position secured, but I was moving countries, so I moved to Sweden for that, and I'd been there a couple of months sort of before everything started to, I know, not fall apart, but, before everything really kicked off with COVID things. And I guess most people are aware that Sweden didn't really take many mitigations. So we were allowed to go to work, but everyone that I worked with very much didn't want to catch COVID, especially at the point where we couldn't get vaccines. So my group, we all worked from home. But when you're new in a country, that can be quite an isolating thing. Mm-hmm. And I ended up having the support of my work colleagues, and they were really great, and absolutely no bad words about them. But it is still very isolating, and very, it was not the experience that I thought I was going to get in Sweden. And then just adding to the, I guess, uncertainty of it all, that it felt like very difficult to come home at that point as well. So
logistically,
with MIQ and everything. Mm-hmm. And in the meantime as well, I'd gotten a dog, so that adds another layer of complexity to an international move. There was a lot to juggle. And then in the end, I sort of decided that the best move for me was to come home. And I was able to, and I feel very lucky that I was able to work for a few months on my Swedish postdoc contract still, with a group that we already collaborated with in Dunedin. So I kind of was able to get that sense of normality back a little bit and continue with my work while I was sort of looking for the next step, because there was the option to continue with my group in Sweden, but I realized that I wanted to be home. I found a way to kind of stay here.
And what was the topic of the research for your postdoc in Sweden?
So that also was sort of, it kind of got turned on its head by COVID things as well. I was supposed to be working on the genomics, like the population genomics of a parasitic nematode, so a little worm that infects reindeer and birds. And we were waiting on sequencing results, but this was all delayed because of the pandemic, because the sequencing facilities were really busy. Plus, a lot of people were working from home if they could and things like this. So it was all a bit slowed down. And then I didn't get the first of the nematode data until towards the end of my time in Sweden as well. So it was really delayed. And in the end, I ended up working on some questions about bird genome evolution, because birds have really conserved genomes. But if you look at bird diversity, there's a lot of it. So there's a lot of questions about, how do they have such a conserved genome, but they're also different to each other.
Wow, that's fascinating. I didn't know that.
That's great. Yeah, I'm still finishing that up, but
Nice. Really interesting question. What is your hunch? What do you think is happening there?
So I was specifically looking at a whole genome alignment of different bird species and trying to look at some sort of different forms of 3D DNA folding based on the sequence and how that was related to conserved elements. And I was kind of surprised, I guess, that these shapes, these DNA folding shapes, were actually not seemingly related to the conserved regions of the genome. I don't know, for some reason I expected the opposite, but I'm still sort of finalizing that work. So maybe there's something else in there that I've overlooked at the moment.
Well, that sounds like really intriguing work. This is just to probe just a little bit further, the 3D shapes of the DNA. This is in the cell? So it's the ones that I was looking at specifically are called G quadruplexes. So they're formed by the guanines of the DNA, so the G that always pairs with the C. And I think there have to be like 4 of them in a row, 3 times or something like that. And then it forms this cube, basically, of DNA, which is really cool. And I'd never really thought about the fact that we don't just have the double helix going on. So. Yeah, that blows my mind a little.
And it
beyond
seems,
the helix.
It seems to be involved in a lot of like gene regulation stuff. It's kind of...
Okay, wow, fascinating. I'll be reading up on that.
Yeah.
So what then happened next? So you finished the postdoc in Sweden, based in New Zealand.
Yeah. Then I actually almost simultaneously heard that Marsden, that I'm an associate investigator on, got funded and got offered the job at PFR. But I got the job offer slightly before I heard about the grant. So that's how I ended up at PFR.
That's so exciting. So that was a Marsden Standard grant?
Yeah.
Right.
Yeah. And so we've ended up hiring someone else on that as a postdoc, and actually also helped her return to New Zealand. So from that side, it kind of feels nice that I was able to do for someone else what I really wanted as well. So,
that's nice. But you are working on that project still?
Yeah, I've
Right.
got a few hours on that.
Yep.
So, yeah.
So you're a postdoctoral scientist now at Plant and Food Research in the seafood production group.
Yeah.
When you look back on your career, what factors do you think helped and hindered your career progression?
That's a bit of a tricky question.
Sorry, that is a curveball.
I don't know. I think I've definitely been helped by the fact that I'm not really afraid to put myself out there too much and I guess move countries, because it's sort of how I grew up as well. I was born in Belgium and my parents moved us to the US when I was 5 and New Zealand when I was a teenager. And actually, when I told them that I was going to go to Europe to do a PhD, my dad's first reaction was like, "No, you're not." And I said, "Try and stop me." What have you been teaching me growing up?
Exactly. Yeah. Bad role modeling.
I think it was just sort of an initial moment of shock that
Sure.
I was going to be so far away and things like that. But yeah, it's all, I think, gone pretty well.
Yeah, it all worked out for the best.
Yeah, yeah.
So that's helped you, putting yourself out there.
Yeah.
Anything that hindered the career progression?
I guess maybe the biggest thing is that I don't come from an academic family. And I've always been encouraged to follow my interest in science and to do well at school, but there's not sort of this understanding of what a PhD is or a postdoc or the career progression or even some of the things that I maybe could have done to sort of support myself or advocate for myself a little bit more in those difficult moments. I just wasn't aware of them. And I think often you can kind of come up against sort of, I guess, very academia unique political situations that might be a little bit challenging to navigate if you don't understand sort of what's happening. Even in places where they say that it's sort of like a flat hierarchy or there's not much of a hierarchy, there is often still a little bit of that. I don't know. It's a bit hard to explain.
I completely
think
relate.
If you don't have that background, it's also a little bit difficult to navigate sometimes.
Yeah, and I guess mentoring is a massive part of helping you navigate that, isn't it?
Absolutely, yeah.
Getting a good mentor.
Yeah.
I completely relate to that experience. So is being a scientist everything you imagined it would be?
Well, I thought I was going to be in the lab all the time, so absolutely not. But I actually really enjoy all the computer-based work, data analysis stuff, far more than I ever thought I could. I never really was much of a computer person, but now I'm coding every day. And I think that's just something that maybe I should have been open to a little bit earlier in my sort of education, I guess. Not my career so much, because as soon as I realized that data analysis could be linked so closely to genetics and genomics, it was sort of just like a puzzle that I could solve, and it was fine and I loved it. But I feel like if I'd sort of gotten into programming and computer things a little bit sooner, I might have had a slightly easier journey at a few stages.
Yes, having some experience of data handling, whatever degree that ends up being, is essential, I think, for a career in science now. Yeah. So I think that if you didn't study coding or some degree of data science at university, there's going to be a big learning curve in there, and you just need to embrace it and get on with it.
Yeah. For me, that was my PhD, and I was the only one at my institute that was really doing a lot of genomics. There were some people doing bacterial genomics, but they were doing a lot of things manually still. And I couldn't do that because I had too much data.
Yeah.
And for my supervisor, it was also the first time doing that kind of project. So we had a collaborator that we worked with, and we visited for like a 10-day crash course on project-specific bioinformatics. But from there, it was just sort of like, let's figure this out.
Figure it out. Absolutely. Well done.
Yeah.
Sounds like a tricky time, which you succeeded in. So how would you summarise your advice to young people thinking about a career in science?
I guess don't be afraid to put yourself out there and learn a little bit of coding.
Learn a little bit of coding, yes.
And just kind of follow what you're interested in, I guess.
Yeah.
And see where it takes you.
And get a good mentor.
Yeah.
Yeah.
That's helpful.
Yep.
Wish I'd done that sooner.
Great.
Yeah.
And thinking about your current research, what questions are most intriguing you right now?
I kind of have quite a few things going on, and I'm at a bit of a stage where a few things are also not going to plan. So there's a little bit of frustration in there. But I think I'm really interested in, down the line, sort of combining a lot of my data sets. So one of the big questions that we have is how to improve things like growth in snapper for aquaculture purposes. And we understand some of the genetics already, and we've been using that to select the snapper for our breeding program. But other things like the environment, so the temperature, the water that they're in, things like that, and their gut microbiome are all things that we're still sort of working on integrating into that. And we know that, of course, if it's warmer, they grow faster, but that's a very simplistic sort of view of that. And it would be really cool, I think, to integrate a lot of different environmental parameters and gut microbiome, community profiles, and growth traits in a machine learning model that hopefully we can collaborate on with our collaborating group in Wellington to sort of figure out the most important factors and how they interact with each other so that we can really start to tailor what the snapper need to do well.
Fascinating. It sounds like you've got hours of computer-based research ahead of you and beautiful puzzles to solve.
Yeah.
Well done. So some exciting news for the listeners. Julie will be joining the team as a new Scigester. So welcome aboard, Julie, and can you tell us about your interest in science communications?
I think I've always kind of dabbled in it, but never had a huge opportunity to really run with it. I sort of regularly tried to explain some of my research on platforms like Twitter in the past, and other than that, it's been challenging because I've lived in countries where I don't speak the language so well. So in Austria, I didn't speak that much German. I did have a bit of a go at it towards the end of my time there. And in Sweden, a lot more people speak English, so it's a little bit easier to sort of get involved in SciComm things. But then, of course, the pandemic came along and sort of scuttled all those in-person opportunities. And yeah, I don't know. I think it's just something I've always been interested in, but not had many opportunities in. So I think it'll be cool to see where this goes.
That's great. I look forward to hearing your episodes.
Cool.
So finally, thinking about the future, what are you most looking forward to?
Yeah, I guess I'm looking forward to seeing where the government's current review of the science sector and the research sector goes, and hopefully getting a little bit more support so more of us can be a little bit less precariously employed.
Absolutely. Yeah, there's big changes coming around with Te Ara Pairangi future pathways. Yeah. Well, thank you so much, Julie. It's been a pleasure speaking with you today. Thanks for sharing your story, and we look forward to hearing more about your science in future episodes.
Thank you for the chance to be on here and interviewing me.
And thank you out there for listening. If you'd like to know more about the topics covered on Scigest, check out the episode pages on the Plant and Food Research website. Please also feel free to subscribe to the Scigest channel and rate and review each episode. Why not give it 5 stars? Thank you, and mā te wā.